Welcome everybody to our final bite-sized science webinar this week. Just give people a few more minutes to dial in. So sit tight, grab yourself a quick cup of tea if you haven't done so already, and we'll get started in just 30 seconds or so. So hi everyone. Stop sharing that screen. Okay, well welcome everyone to our final bite-sized webinar of the week, celebrating National Science Week. We're delighted to have you with us. And by us, I mean Kate. I'm the Community Engagement Coordinator for IHMRI, and Dr. Yee Lian, who's going to talk to us about her research today. So get your questions ready. So due to COVID, like a lot of organisations, we've had to take our events online, which is why we're doing these webinars. And we're calling them bite-sized webinars because they're designed just to give you a little taste of the types of research that we do here at IHMRI. IHMRI is a not-for-profit organisation, for those of you who don't know. And our goal is to support health and medical researchers in the Illawarra-Shoalhaven region by bringing them together with health professionals, doctors, in order to solve our greatest health challenges. And we do research into all different types of diseases, from cancer to diabetes, mental health to dementia, just to name a few. I just want to say a really big thank you to all of our staff, researchers and students. I know the last few months have been really quite challenging, but I just want to thank you for all the hard work you've put in, taking your teaching online, and also just all the effort that you've put in to ensure that your research can continue during this time. So we really do appreciate your support. Thank you. And of course, thank you to our community groups and individuals who support IHMRI. We couldn't do our work without you, so thank you so much to you as well. I'm sure lots of you are dialing in today. So on to our webinar. If you have any questions, please pop those in the Q&A box. And if you have any comments, you can pop those in the chat. And while I'm mentioning the chat, if just a few of you could just type in there to let me know that you can hear and see us okay, that would be wonderful. And we'll also have a survey at the end of the... Okay. So I haven't seen any... Can you all hear us okay? Oh, wonderful. Thank you, Debbie. Lovely. Thanks, everyone. Okay, so I think that we will get on with the webinar. So a big welcome to Dr. Yee Lian Yee Lian has spent the last 9 years of her life trying to understand life through the worm. She did her PhD at the University of Sydney and then moved to Cambridge in the UK to the Laboratory of Molecular Biology to study worms in colder weather. You definitely moved to the right place. She's currently at the University of Wollongong and Illawarra Health and Medical Research Institute, leading a research group with the goal to understand the signals that allow the networks in our brain to learn to adapt to changing environments. Welcome, Dr. Yee Lian.
Thank you, Kate, for the introduction, and thank you everyone for coming. Kate, could you please share the poll?
Absolutely.
So I just had a little icebreaker activity for all of us. Can everyone see that poll? It's just 3 questions, and I invite you to put your responses in. Don't worry if it's wrong, it's fine, or you're not sure. Have a guess and have a go. So these questions are about how many neurons do humans have, or a bird, such as a pigeon, or a nematode worm. And in case you're wondering what a nematode worm is, it is this. So, so a nematode worm is a very tiny, it's a very tiny microscopic animal, and surprisingly, perhaps, it is a great model for neuroscience research. So has everyone popped in their responses to the poll? Because we need to see the results before I start my presentation. Okay.
Okay, so I'll give you 10 more seconds. We've got 5, 6, 7 results.
Just click
Just a few more.
Use your instincts.
All right, I'm going to end the poll now.
Perfect, thank you. So let's have a look at the results of this poll. All right, so, okay. So humans, so I'll just tell you the answers. So that is correct that humans have 100 billion neurons, which may sound like a lot to us, because 100 million is, sorry, my bad. I'm actually misspeaking. It's 100 billion neurons. 100 million is in a house mouse. So 100 billion neurons in the human brain, which sounds like a lot, because it is a lot. It's a number that we can't actually fathom in our minds. So perhaps 100 million sounded like something a bit smaller and more accessible, but it is true that we have around 86 billion to 100 billion neurons in the human. For the second, how many neurons does a pigeon have? So we all have seen pigeons. They may not seem like the most intelligent of animals, but they do have 300 million neurons, so option 1. So yeah, perhaps that is a little bit surprising again, that pigeons have more neurons than you would think. Perhaps that does not translate directly to how intelligent the birds are, but that's how it is. And number 3, how many neurons does a nematode worm have? So congratulations to 50% of you who have probably heard me say this before many times. The worm has 300 neurons. So just going down from the human with 100 billion neurons to the pigeon, which has 300,000, to a worm, which has just 300. So we can see that, different animals have different numbers of neurons, and perhaps it's not so straightforward to determine how smart an animal is based on how many neurons they have. So I'm just going to start sharing my screen right now. So yeah, if you have any questions about any of this, please feel free to pop it into the Q&A. There are no stupid questions. So, we're all here to learn and discover, and to be excited about science. So I'm going to be talking about how we can learn about the brain through the worm. And as I've mentioned, when I talk about worms, I don't mean earthworms, I mean nematode worms, which is like this guy. So I'm just going to change to my pointer. So this is the head of the worm. So we're seeing some of the neurons in the head, and that's the tail, and these are its little eggs through which its babies hatch and become adult worms. Okay, so let me tell you a little bit about me. So how did I become a scientist? So when I was a little kid, my parents can attest to this if you ever managed to talk to them. I asked questions about everything. I was curious about every single thing in the world. Is the sky blue? Why is it blue? How do telephones work? Why does poop smell? Why do people look like that? Why is hair color in this way? And I guess I never stopped asking questions. And at some point I realized that I could develop the tools to answer some of those questions. So in 2010, I graduated with a Bachelor of Science from the University of Sydney. And then I went on to do a PhD. So I graduated with my PhD in 2015. And after this, I moved to Cambridge in the UK to do a postdoc. So, you may think of Cambridge as this, really fancy place with all these old buildings. I wasn't in one of those. I was actually in something like this, which is perhaps a little bit less exciting in terms of beauty, but it certainly had a lot of great science going on. And then I returned to Australia in 2019 to the University of Wollongong and IHMRI, where I'm now based in this very fancy new building. And please do come visit. Okay, so that's about me. So my research is about looking at the brain. And when I think of the brain, I don't think of it as kind of a mushy mass of tissues. I think of it as kind of a electrical circuit, like a wiring diagram. So if you imagine a circuit such as this, so a very, very simple electrical circuit, you could imagine that if everything was working appropriately, all the connections are there, the cells are connected to each other in the appropriate way, and all the signals are going the way you would expect, you have a healthy, functioning brain. Excellent. So what if something goes wrong? What if one of these connections is broken? Then we may have a defect in the brain, such as memory loss, dementia. what if we have some of these connections overactive, overstimulated in this circuit? What could happen then? Well, we might have another defect in the brain. It could have going the other way, such as chronic pain and anxiety, where some connections are overly stimulated in non-physiological, not normal conditions. So this is kind of how I see my research. But of course, when we are looking at circuit diagrams, we need something that is fairly simple. We need something that we can manipulate, something that is accessible for us to understand in simple terms. So if this is the brain of a simple circuit, and the human brain has 100 billion neurons, then the circuit of the human brain would look something like this, right? So, okay, this is actually a circuit diagram, I think, of a motorcycle, but you kind of get the idea. So we want to be able to study simple systems in order to translate our findings to more complex systems, because these complex systems are really very difficult for us to access experimentally. So the goal of my research is to understand the wiring diagram of the brain so that when we are able to fix the circuit when things are broken. So that's the overall goal. So how do I do this? So to solve brain circuits, I need a model system. So I use this nematode worm, which has a very long name called Caenorhabditis elegans, which looks a bit like this. So although it looks quite big on my screen, it's actually just 1 millimeter in length. It's a tiny nematode worm. And we use it for a variety of reasons. Firstly, it's very genetically accessible. So we know a lot about its genetics, which means we can manipulate it and change certain bits of the genetics so that we can ask questions about which genes are important. They're really easy to maintain, and I'll show you what this means in a second. And they have a really compact nervous system, which consists of 302 brain cells. Importantly, something that is super cool about the worm is the fact that they have such small brains means that we know the exact connectivity of all of its neurons. And this is something really unique. So we know exactly which neurons are connected to each other. And this is a tool that is fairly powerful when we're studying the circuit diagram of an animal. And perhaps you may not think that this little wormy can tell you much about people, but there is a surprising level of similarity to higher organisms, where 80% of the genes in the worm's brain are actually very similar to the genes in the human brain. So what do I mean when I say the worm is easy to maintain? Well, how do we grow the worms? So because they're not earthworms, we don't grow them in the earth, as many people do tend to ask me, but we grow them on agar plates, which are as shown here. So we pour lots and lots of these small agar plates and we feed them bacteria. So I don't know if you can see these like little textural bits on the plate. So these are the worms on the plate. These are their eggs and these are their larvae. And they're just growing on bacteria. So we grow the bacteria in the lab. Bacteria is non-toxic, non-hazardous. It's not dangerous to any of us. And the worms just love it. They just eat it up. So that's their main food source. So this is fairly cheap and easy for us to grow. And we can grow lots and lots of worms, as you can see from the number of plates we have. So why else do we use the worm? Like I said, they're very genetically accessible. So we have a variety of tools that we use to solve our model brain. We look at them from the genetic perspective. We can look at cells and circuits of the brain, so like this. And then we can study their behavior. So let me explain a few of the tools that we can use. So firstly, when it comes to genetics, so this is the germline of the worm. And what we do to manipulate its genome is we can inject what we call transgenes that can be then taken up into this germline, and the worm just makes the products of those transgenes. It's very simple. It takes just a week or so to make a worm that will generate those transgene products. And now with the advent of CRISPR-Cas9, I suppose some people might know what this is. It's a form of genome editing. We can then basically make any type of genetically modified worm that we would like for scientific reasons. So we don't do it for fun. We do it for a targeted scientific purpose. But this convenience of accessing the genetics is really powerful in this model. The next thing we can do is optogenetics. So this means light plus genetics. And the way this works here is we put in a molecular switch in specific cells of the brain in the worm. and we can then activate that switch using light. So in this case, we use a blue light. We shine it onto the worms, and we see what happens when we activate those neurons. So I'm just going to play you a quick video of when we activate the neuron, which is responsible for an escape behavior. So the worm is now crawling. When you turn the blue light on, it reverses, right? So you can see it responds basically immediately. So this tool is very powerful because we can then ask, what do specific neurons do in the worm brain? We can just put this molecular switch in, turn on a blue light, and see its response. Okay. We can also do optogenetics in groups of cells. So in this animal here, we have put the molecular switch in neurons that make dopamine. So if we turn on the blue light, as of now, the worm immediately slows down. So this means that if we activate the neurons that make dopamine in the worm, this leads to an immediate slowing response. So we can then start to ask questions about how that occurs at the cellular level or at the molecular level to kind of connect that to the behavior. Okay, and another cool thing we can pair this with is recording neural activity. So this video sort of happens kind of quickly, so I'm just going to point out where you should be looking. So up here, where my pointer is showing, these are two neurons, and the arrow is pointing at the neuron that you should be looking at. So what we've done is we've put in little sensors for neural activity in these cells, and they will light up when the cell becomes active. So just watch. Yay. So, and we can then pair this to specific stimulations. So we can poke the worm, we can wash over some chemicals, we can ask at which point does this neuron become active? So this presents a whole bunch of possibilities for us. Okay, so now that we've gone from genetics to looking at the cells, and now we're going to look at behavior. So these are worms crawling on a plate, and the little colored lines are just tracking their movement. So what we've done here, it may look a little bit cruel, but don't worry, they're not hurt. We're just poking the worms with a little metal pick, and we're watching them kind of dive away from the pick. And now they're crawling away at high speed. This is sped up. And we can then look at these lengths of these colored tracks and quantify them. And this will let us know if specific changes in the worm's genes, for example, can affect their ability to move away from this scary metal pick that comes from the sky. And this combined with video tracking and automated analysis means we can do lots of these experiments very quickly. Oh, and another behavioral test is this touch assay, where we kind of gently stroke the worm. So the head is up here and the tail is down here. When we stroke the animal and the anterior on the front side or the back side, it will usually accelerate in the opposite direction. And through understanding this touch circuit, we've been able to figure out quite a lot about how the connectivity of the worm touch neurons actually connects to the rest of the brain, which is super cool. Okay, so I've got another video here. The sound will not play. Oh, no. So I noticed sort of during my, okay, well, maybe you can just listen to the music. So if you put a drop of this liquid onto the worm, sorry for that. If you put a drop of liquid onto the worm, it will reverse. I realized that my colleague had inserted music into this video only when I started to play it at an international conference. So I apologize that I forgot to turn it off this time, but anyway, that's how it is. But if you put a drop of this liquid onto the worm, which is a repellent, so something that the worm doesn't find particularly friendly, it will reverse. And that is the behavior that we can record here. Okay, so moving on, another thing, a behavior that we can detect, for example, is a variety. So we can detect a variety of behaviors through video recording. But another thing that is very interesting is that if the worm does not have dopamine, as we saw earlier, dopamine is an important signal to slow down the movement of the worm. If the worm does not have dopamine, it responds differently to the presence of food. So in case people don't know what dopamine is, it is a neurochemical which is released by particular cells of the brain, and it acts in a sort of hormonal fashion. So we can activate parts of the brain which are a little bit further away from where it was originally released. And dopamine is very important, it seems, for in humans as well, for our reward-based behaviors and our motivations. So you may have heard of dopamine before, but it's perhaps maybe surprising that worms also have dopamine, and they are also important for slowing down a response to food. So in this animal here on the left, it's what we call a wild type animal, so it's not a mutant. So it's coming into a food patch right here. And you can see its head moves in, it's exploring, just wriggling around, and it's slowing down as it goes into the food. So on the right side, we have a dopamine mutant, so dopamine negative. So these worms don't have any dopamine. And what they do when they come into the food patch is basically nothing. They're like, oh, nothing is there, there's no food, I'm just gonna keep moving with my life. So we can then use these behaviors to investigate what's really important with dopamine responses in the worm. Okay, so just before I move on to my own research, show a little bit of how we build the robots that actually do this work. So this robot here has eyes, which are completely unnecessary for the actual work, but it just looks cute. And this robot kind of taps the plate on which the worms are on. And it means that they respond to the tap either by reversing or accelerating in a forward direction. And we can analyze all these behaviors and link them to the molecular and cellular changes in the animal. So although this may all sound quite fundamental in terms of what happens in the brain, we can use this to translate to to diseases or conditions in the brain which are affecting humans, because the worm is essentially a smaller and simpler version of the circuit diagram that's in humans. So part of my research focuses on chronic pain, which is a condition that affects up to a quarter of people in the world, with a higher prevalence in older people. Current treatment strategies, which involve opiates, are not really effective in quite a lot of patients, and they have pretty horrible side effects, such as addiction, which is contributing to about 30% of drug-induced deaths in Australia. So it's pretty clear that we need a better way to treat chronic pain. And one way we might do this is by looking at a very fundamental process in the brain, which depends on the, which is called sensitization of pain receptor cells, where basically these cells, with every time that they start, every time that they start doing something, becoming activated, the next time they become activated even more. So we use our research and all the tools that I've shown you earlier to identify a specific neurochemical pathway which is required for this pain sensitization. So we have this neurochemical called FLIP20, which we identified using this behavior test I showed you earlier. We also have this receptor, which we identified, which we identified through these neural activity recordings I showed you earlier, and then we can show how they affect this pain receptor cell. And overall, we showed that this neurochemical pathway is able to trigger sensitivity of this pain receptor cell. What we're going to do is use this paradigm to investigate the basis of chronic pain in a simple system and in a way that can be translated to humans. Okay, so just before we move for questions, I hope there are lots of questions. I'd like to acknowledge the people in Cambridge who were really involved in this work, as well as my current team at IHMRI and UOW School of Chemistry and Molecular Bioscience and the Molecular Horizons Research Institute. So this is my team. They're not, that's me when I had shorter hair. They're not all freakishly tall. I'm just quite short. So yeah, that's us. And you can check out our lab website here. And I'll just show you some pictures of the lab. So we use a lot of microscopes. We use a lot of colorful plastic boxes apparently. And this is an example of how we look at fluorescence in the worm. And with that, I'll leave you a picture of fluorescing worms on a plate. So each of this is an individual animal. And I thank you very much for your attention and open for questions.
Fantastic. Thank you so much, Yee Lian. That was really, really interesting. love all the commentary on the worms and what they're up to. So we do have some questions for you. First up, will the new electron microscope, so I'm assuming that's the one in Molecular Horizons, how will this help you with your work, and what other equipment do you use?
Yeah, so we don't directly use the cryo-EM, as it's called, but we do work together with those colleagues to basically understand the shapes of the proteins that we're working with. And it's actually quite a good question, because we can use the worm to make lots and lots of these proteins, which are these small molecular machines, and we can then look at their shapes and their structures using the cryo-EM. So I think in future we will work very closely with people who are using that microscope, and hopefully we can make pretty pictures of molecular machines together.
Great. It's a great question. Can you tell one worm from the other?
So no, don't tell my worms that. But,
don't have any favorites.
I know, no, don't tell them this, but they all do look relatively the same.
Mm-hmm.
Certain genetic mutants or transgenic animals, like the fluorescing ones, they do look obviously different, but if they are siblings and genetically identical, I'm afraid they all look pretty much the same. Oh. I do name them though. Yeah.
Every single one of them?
Sometimes.
No judgment. Oh, Yee Lian. How big are the worms?
So the worms are 1 millimeter in length as adults. So as baby worms, they are significantly smaller. So we have to use a microscope for everything. And all of my students do complain about the time it takes to learn how to gently pick a worm, a tiny worm, under a microscope from one plate and move it to another plate, because it requires some fairly advanced fine motor skills.
Great. Somebody here wants to know the robot that you, the picture of the robot that you showed in your slides. What job do they have? Do they actually replace the work that a person would do?
So yeah, so there's no replacement for people. When people have to do the experiments and people have to think of the scientific questions to actually ask, the thing that the robot does is it allows you to do things either faster or without supervision. So the kind of tapping robot that I showed earlier, it just means that every time you tap the plate with the robot, it's essentially the same. Whereas if I was to go and tap the plate myself manually, there might be slight differences every time I do it, even though I'm trying to be really careful. So the robots are helpful, but they can't really take over our jobs. Not at the moment. They're not that smart.
How long does a worm live? And how long does it take to grow a worm to the stage where you can do experiments?
Yeah, so that's a really good question. So the worms take about 4 days to go from the egg to the adult that can lay eggs. So that's not that long. And in adulthood, they live for maybe 2 or 3 weeks. So yeah, it takes 4 days for us to be able to do experiments on them, and 2 or 3 weeks for them to live, and so we can do lifespan experiments. Something I didn't mention is that the worms are hermaphrodites, so they self-fertilize. They have both male and female germlines, gonads, so they kind of fertilize their own eggs. So they just need themselves. One worm makes many clone babies.
And I guess that helps then in terms of when the 3 weeks after that lifespan of the worm, so you just have an endless supply of worms at your disposal.
That's basically an endless supply of worms, that is correct. Makes me very happy.
We've got a question here. Where and in which kind of treatment is sensitization of pain receptor cells applied? Yeah,
so that's a good question. So most of the time we use optogenetics. So we put this molecular switch in a neuron which we know senses pain, and we can then turn on that neuron with the blue light signal. So that's the treatment that we use. And so the idea is that once we get good at doing this in a more high throughput, so lots more experiments quicker, we can then use this to apply perhaps pharmacological interventions, so drug interventions, to see if this improves or decreases the response.
so I think is it Tim? I think that asked that question. Oh. They've just asked, and can this be applied on humans?
So there is some talk about using optogenetics in humans, but because you have to shine a light onto the neuron, that is kind of difficult in people because we have a skull and skin, whereas the worms are transparent. Yeah. So, so we can't do pain studies in the exact same way that we do pain studies in, say, a mouse, but we do them slightly differently in the worm. We kind of activate that pain neuron directly.
Okay. Got it. I have a couple of questions. I know we're just on time, but I would love to know, Yee Lian, what you like most about your job as a scientist.
Oh, I think it's the thrill of discovery. I mean, it's most of the time it's so when you do an experiment, 99% of the time it doesn't work the way you expect. But instead of making me sad, I think that's actually really exciting. For a moment when you discover something in the lab, you are the first and only person to know that thing. And it's kind of this exciting idea of being able to use that knowledge for good. It's the best part of my job, and also to train the students who are then learning to use science in their lives. That's another really awesome part of my job.
Yeah. And I guess when experiments don't go quite right, then it generates a whole load of other questions for you to then go about answering.
Yeah.
One more question, I think, if you weren't a researcher, Yee Lian, what do you think you'd be doing with your life?
That's a very good question. I ask myself this every day. I think I would probably be a baker, but not a very good one. I think I would just bake to eat. And it's because, people say baking is science, and it really is. It really is, and I really enjoy it. And I also really enjoy the after part, which is the eating.
Yeah, I'm sure all your friends do as well. Assuming you share your baked goods with.
Not recently, there's a pandemic.
No. And has COVID affected your research?
Yeah. I think we are affected, like everyone is affected. Not so badly. We can go back into the lab now, and it's been great to see the students and catch up with everybody. But we do have to bear in mind that to keep everything safe, we have to maintain physical distancing. We have to wear masks. We have to put on more protective equipment. So it's different. And during the times we had to stay at home, that was hard. But we still kept in touch. There's the internet, so we Zoomed every week to make sure everyone is doing okay. Yeah. But yeah, I think most of all, we just have to take care of ourselves and each other, and be kind, because it's tough times.
Very good message there, Yee Lian. Well, look, your passion and your love for your worms and the work that you do and answering all these questions is incredibly clear. And Yee Lian, we're just so grateful that you were able to spare some time to share your research with us. So thank you so much. Thanks to everybody who dialed in today and for asking the questions. I know there's so many more questions that we all want to ask. So watch this space. We're hoping to do a few more webinars before the end of the year. I don't think webinars are going anywhere. I feel like this might become the norm. So keep an eye out for more ways that you can hear from Yee Lian and the research that she does. So thank you so much, Yee Lian.
Thank you, Kate, for organizing all of this, and thank you, everyone, for attending. I'll see you next time, and email me if you want to.
Actually, what's your email address, Yee Lian? Would you share that in the...
Ooh, I don't know, maybe. YLCHEW. But also, if you have Twitter, I'm on Twitter a lot. And my Twitter name is Wormy Chew. Yes, it is.
Wormy
Chew. The way you would
think. Of course it is.
Yeah.
Okay, that's great.
Yes. Yeah.
I'm going to share a screen as well with our IHMRI details. So you can go to Twitter at IHMRI, facebook.com/IHMRI to find out about more webinars that we're having down the line. And every day this week, one of our lab teams has taken over our social media. So there's lots of diverse and exciting content up there. So I really encourage you to go and have a look at that. And of course, if you'd like to support IHMRI, you can go to ihmri.org.au/donate. We are a not-for-profit organisation, and we're incredibly grateful for any donations that you might be able to make to allow us to support our amazing researchers. Do get in touch if you have any questions, but for now, I will say goodbye, and thanks again to Yee Lian for sharing your research with us.
Thank you. Bye.
Bye, everybody.